It's my great pleasure to be here to introduce today's talk. The series of talks that this is a part of is, I think, probably one of, Andy's not here, so I can say it's probably one of the best things that the IAS Center does. It recognizes the importance of the Nobel Prizes that are awarded every year, and draws on the expertise of our own faculty to help explain the prize and explain why it was chosen as a prize and why it's so important, not just to the one field in which it's awarded, but to the larger field of science as a whole. So this is the third of this year's series, and today we're going to hear about the 2017 prize in Physiology or Medicine that was awarded to the three gentlemen behind me for their work on circadian rhythms. And as much of the work on this that led to this prize was done, although it's a prize for physiology and medicine, which typically implies human physiology and medicine. Despite that, much of the work you're going to hear about was actually done in the fruit fly. And we are fortunate enough in the life science department to have a really superb fruit fly geneticist who is going to be your guide for today's talk to the prize. So our speaker today is Yan, who joined the HKUST faculty not that many years ago. But as I said, she's an expert in fruit fly genetics and so well positioned to give this talk. Her bachelor's degree is from Beijing University. She then went to the United States for training first as a PhD candidate, a doctoral candidate at Princeton University, and then moved to the other U.S. coast and did a postdoctoral fellowship with Chris Do in Oregon. And I think enough said. I'd like to now turn the dais over to Yan, and please join me in welcoming her while we learn about this year's prize in Physiology or Medicine.
Okay, thank you, Carl, for the introduction. So let me see. Okay, so the circadian rhythm, or this innate sense of time, is not something that we are unfamiliar with. For example, now it's 3 p.m. Within the next hour, many of us will feel pretty sleepy. Now, part of it may be because you find the topic is not as interesting as you thought, but partially it's because your innate biology clock is telling you it's probably time for a little siesta. So let's see. But the first scientific experiment documented or published was in 1729 by this French astronomer, de Marin. What he did was to put touch-sensitive plant into a dark room. So this plant will open its leaves in the morning and fold its leaves in the evening when sun goes down. There's no way we can know what kind of which species of plants he used, because as you can see, publication at the time is a very different game. You can see this is the only sentence about the materials and the method. He said this experiment was carried out in the end of summer and well duplicated, so nobody really knows which species of plants he used. But he did put the plants into constant darkness, and plants keep opening its leaves in the morning and folds its leaves in the evening, as they would do in normal environment. Now, this experiment actually inspired a lot of experiments. So people have tried to put this type of plant into deep in the wine cave. So as you can imagine, in a dark room, the temperature or humidity control is not very good. But if you put it down into a wine cave, not only it's dark, the temperature and the humidity wouldn't fluctuate so much. Now, by 1960s, people start to design experiments such as taking potatoes to South Pole and see whether that will change the potatoes' circadian rhythm behavior. Now, and then they even proposed to take potatoes to the space, to NASA, and see whether orbiting in the space will change the circadian rhythm for potatoes. But nobody, so by that time, it's pretty clear that all the living organisms process some sort of innate sense of time. It's not a pure response to the night or dark cycle. It's something we're born with, and it's very much intrinsic. But nobody knows what controls this intrinsic clock. And here, Seymour Benzer and Ronaldo Colombka published the first paper about the assassination of clock mutants in Drosophila in 1971. Now, Drosophila by that time is not a new research organism. Drosophila rise as a research organism in 1909 when Thomas Morgan published the white gene that started modern genetics. Now, so I see some of the audience a lot familiar with Drosophila, so I will play a very short video tape. This clip, this is a restoration of the original fly room used by Thomas Morgan. You can see, let me see if I can play this. You can see flies really love bananas. So if you have leftover fruits in your home, 99% chance you will find Drosophila. And fruit flies are kept in those bottles, or in the old days, it's milk bottles. And legend has that Thomas Morgan is very cheap, so he actually stole milk bottles on the street. These days we use plastic ones. Now you can see soon, let me see. You can see those labels here. So each bottle is an independent genotype. So you should never mix bottles of flies together because each of them usually contain a very distinct genotype. Now soon you can see flies that are very, very small. These guys here, they're feeding. So they do have behavior. How they feed, how they do courtship, those type of things have very stereotypical behavior. And you can see a larvae here. Adult flies come from larvae that turn into pupae and then flies. And flies have a lot of markers. You can see here the wings have different shape, the eyes have different shape. Actually, for each single bristle on fly has been studied and counted. So, and you can see, to work with flies, you have to put them into sleep. In the old days, it's a very old-fashioned method of using ether. So this is, we don't use ether anymore because it's actually, it smells. So we're using CO2 to put flies into sleep. I gently tap them on the fly pad. And now, soon you can see the males and females under the microscope. As you use a gentle brush to deal with flies. This guy here is a female. So it's going to be more clear here. You can see their eyes are red, they have wings, and this is a virgin female, and this is a male. And so this is how so not much has changed in terms of how to handle flies over the past 100 years. Now, so back to circadian rhythm. So the person who started all of this, Zemo Benzer, he's not a Drosophilist. He's not an academic descendant from Thomas Morgan. He actually started his training as a physicist. And he actually did pretty well in physics. He held 6 patents. He probably worked on some military-related projects, so he's very well funded. But he decided he was more interested in biology. So he switched to work in the phage group, the famous phage group who figured out the whole gene structure and stuff. Now his work on the R2 gene mapping actually won him a NASCAR award in 1971. But by mid-1960s, he started to get interested to behavior. In an interview archived in Caltech, he stated about his reason why he decided to switch from molecular genetics to behaviors because he has 2 children. And when he watched the 2 children who were genetically related, they actually have very different behaviors. So he started to think what's the difference? Is that the genetics that determine the very different behaviors for these children? Now, being Benzer, it's not very surprising that he decided to address or dissect the behavior problem using a genetics approach. Now, if you go back 50, 60 years ago, people who study behavior are not thinking about genes or the molecules that underlying the behavior. There's always this debate between nature versus nurture. But Benzer decided to adopt a genetic approach to this problem. Now, also, at the time when he moved to Caltech, he met Edward Lewis. Now, Edward Lewis is a real descendant from academic descendant of Thomas Morgan. So he has been working with flies his entire life. Now, but at the time, in 1960s, flies are not very fashionable. It's not a very fashionable organism. So even Benzer thought that flies means something that's very, very much old-fashioned. But obviously, he was wrong because later, Edward Lewis actually won a Nobel Prize for his study in the genetic control of embryonic development. So I'm actually more familiar with this type of study because, but that's a story for a different day. So with the help of Ed Lewis, he was able to set up a fly lab and starts to mutagenize flies to isolate behavior mutants. So his first paper was published in 1967. So this is a paper studying the phototaxis behavior of Drosophila. So it's more like a proof of concept paper or a mission statement for him to show that you can really mutagenize flies and isolate fly mutants with behavioral defects reliably. So, but his real breakthrough comes from the study of his first graduate student, Ronald Klomka. The student was interested in circadian rhythm, and he started his work on flies and on plants, but later switched to Drosophila. And his PhD project was to mutagenize flies and isolate mutants that affect circadian rhythm. So this is the first paper published in 1971. And in this paper, they did a mutagenesis screen. They fed flies with mutagen established independent mutant line, and then assay for circadian rhythm behavior defect. So what they are assaying for is the incursion time of Drosophila. So flies, just like butterflies, they come out of pupae And butterflies come out of cocoons. That's probably when you read the Eric Connor's Caterpillar book, then you know that butterflies will come out from the cocoons. So flies also come out from the pupae or cocoons. And they come out every morning around 7 a.m. And so flies, the name Drosophila actually is dew gnawers. They gnaw to come out of pupae in the morning and drink water. So you can see this is a wild-type fly. They come out every morning regularly. Now, in this mutant, this is the first mutant of Chromcast screening. And you can see the flies are coming out at random hours or throughout the day. And he also isolated another 2 mutants. So these mutants are also coming out at regular intervals, but the period of those flies are different. So normally these guys come out every morning at 7. Now these guys have a shorter period. So if they come out this morning at 7, tomorrow they're going to come out at 2 a.m. And this is a short period mutant. And this is a long period mutant where the flies will enclose at a period of about 28 hours. So this is the first assay for circadian behavior defects. Now, the next experiment they did is a very simple locomotor activity assay. And they actually designed a very simple but neat device to look at this. So they put one fly into a tube. The fly would walk along the tube if they are awake. And whenever they block the infrared light, they will leave a signal on the detector. So if flies are alive, awake, you will detect a lot of signals. If flies are sleeping, then you will see a line like this without signal. So again, this is a wild-type flies. You can see they're pretty active during the day, and they will be sleeping during the night. Now, note that actually in the afternoon, flies also take siesta. So for the first mutant that come out at random hours, they actually have a very random local motor activity pattern. They are actually awake and sleep at random hours. Now, the second and the third mutants, the short period mutants and long period mutants, they do wake up and sleep regularly, but at a shorter or longer period. So those are the first 3 mutants isolated from Chromcast genetic screening. Now, being Drosophila, they do have some genetic tools to map those mutants. So Chromcast mapped those 3 alleles to the same locus on the X chromosome. So since they are mapped to the same locus, it suggests that they are the different alleles of the same gene. Should he work on other mouse, not particularly on mouse, like pig or rat, it's going to be very hard to do this type of mapping because he was working on Drosophila. Drosophila already have 50 years of genetic tools accumulated to do this type of fine mapping. But the problem at the time, this is 1971, the modern molecular techniques are not available yet to do the cloning. So it wasn't until middle 1980s, the Nobel laureates of this year managed to clone the period gene. So Jeffrey Hall actually, he was a postdoc with Zemo Benzer, so he was aware of the clock mutant. And Michael Rosbach was a colleague with him in Brandeis University, and he actually have the molecular training to make this cloning this gene possible. And Michael Young started this now study independently. So in 1984, these 2 competing groups published their first paper on the cloning of the period gene. So what they did is conceptually very simple. What they did is to cut the DNA fragment from the X chromosome around that locus and inject the DNA fragment into the per mutant, and then look for transformants carrying the DNA fragment that rescues the per mutant. So you can see here again, wild-type flies, they come out at regular intervals every day. And per mutants, still you can see this random inclusion behavior. And in this mutant, this DNA fragment here more or less rescued the per mutant, so that now per mutants are coming out of the pupae at regular intervals and mostly in the morning. So the next experiment, again, is using the same locomotor activity assay. So here actually C is a wild type. So you can see flies are pretty active during the day and pretty quiescent during the night. And in per mutants, this regular pattern disappear. Now, if they put this fragment back into per mutants, this pattern is more similar to the wild-type flies in comparison with the per mutant flies. Now, they are able to deduce, because they now got the DNA fragment, they're able to deduce the sequence of the gene as well as the protein sequence. And you can see here, the mutant that doesn't have any circadian rhythm, the mutant that encodes at random hours, it's a mutation that needed to protein truncation. So that's very likely to be a long mutation. And the two mutants with longer or shorter period, they carry amino acid change in their protein sequences. So this was 1987. Now, so let's review the timeline. The mutant was isolated in 1971, and they were cloned between the period of 1984 to 1987. But after that obvious question, what does period gene or period protein do? Now, they actually, during that, the following 5 years, they actually made a lot of mistakes. They thought period gene may be a gap junction protein. They thought it may be an extracellular matrix protein. They published their paper in Nature and other journals that the Nature have to retract. So period become this gene that's, well, promised, well, there's a quote from the Drosophila community, it's a gene that promised so much, but really never quite delivered. And this was 20 years before people figure out what period gene does and how important that this gene is. And during this time, Klompka failed tenure in twice. First time in Caltech, second time in Clarkson College. And our Nobel laureates this year, they persisted over this long struggle. Now, so the breakthrough comes in the early 90s when Michael Rosbach and Jeffrey Hall's postdoc or graduate student, I forgot this one, Catherine and Savicki, she decided to cut the fly head hour by hour and check the protein level of period. At the time, they also got a very good antibody to look at period protein itself. And they noticed that actually period proteins oscillate over a 24-hour, both in the neurons, the photoreceptors, and the glia cells. And other students following the same using the same strategy of check the protein and RNA expression level every hour in the flies, and he also noticed that the per RNA oscillate. So both the RNA and the protein oscillate on a 24-hour cycle. Also, there is a phase shift between the oscillation of the RNA and oscillation protein. So that's, based on that, they are able to propose a working model that if a per protein function as a inhibitor of its own transcription, that in principle would be able to create oscillation like this. And this model turns out to be true, but later proven experimentally to be true. At the same time, the other Norrid, Michael Young's group, was continuing the screening strategy started by Benzer. So they continued the screening and isolated the more clock mutants. The second clock mutant they isolated is called timeless. Again, you can see the timeless mutant, instead of coming out at regular intervals, they come out at all random hours. Again, this is a motto. By this time, you will be familiar with this as type of assay. You can see wild-type flies, they are active during the day and they are sleeping at night, and timeless flies, they don't have this sleep-wake cycle. Now, and also similar to per, timeless also oscillate. You can see here the timeless RNA is cycles over a 24-hour cycle. So by now they have two mutants. The mutant of those two genes lose circadian rhythm behavior. And moreover, these two genes both cycle at or oscillate at their protein and RNA level. So now they have they're better equipped to dissect what those two genes are doing. So now they found that period gene actually localized to the cell nucleus, indicating that it can be a transcriptional regulator that regulates gene expression. And they also found that in timeless mutant, a period actually failed to localize to the cell nucleus. Here, actually, it's a little bit hard to read from the original paper, but here this is the photo receptors of this of flies, and you can see the dark brown staining or the nucleus staining of the period protein. And in the mutant here, in timeless mutant and the per mutant, this nucleus staining is lost. So together, this type of study really opened a new research field of circadian rhythm research field. And by now we know that per period protein and timeless protein, they functioning together to create this lactive feedback loop to repress its own gene expression. So during the day, as the protein accumulate, it will goes in, once it passes a certain threshold, it goes into the nucleus and turn off its own expression. And then as the RNA level decay, protein level decrease, then the transcription will start again. So this is how how the circadian rhythm circuitry is regulated. Obviously, there are a lot more studies about how this circuitry can be fine-tuned. Now, all of this in flies are very neat. So, what about mammals? Is there a similar clock system in mammals? And shown here is the first in 1994, Takahashi's group estimated the first clock mutant in mouse. And he actually adopted the same strategy as Benzer. He fed a mouse with a mutagen to generate mutant flies and looking for mutants with abnormal circadian rhythm behavior. Again, this you can see this the more or less a normal mouse. They actually have a regular sleep or wake pattern. And in the mutant mouse, you can see their sleep wake pattern is completely destroyed. Now, it's worth noteworthy that at the time, actually, many people tried to clone the homolog of period in mouse. But because of the relatively limited homology in the whole sequence, they're not able to find the exact homolog of period at the time. And so actually it's a more brutal force mouse screening strategy that worked. But once they cloned the clock, the clock gene in mouse, they actually found its share important homology in the most important domain in clock gene and the period gene. This is a path domain in the clock gene and the period gene. Now, how about in humans? Now, similarly, in humans, people have found that per mutation lead to circadian rhythm behavior defects. So this is a family with a familiar advanced sleep phase syndrome. So these people, they will go to sleep around 7:30 p.m. and they will wake up around 2 or 3 a.m. And when people checked their genome, it's found that their PER gene, the human PER gene, carry a phosphorylation mutation in its sequence. So now this is a simplified version of the mammalian clock. It looks a little bit complicated, more complicated than the clock in flies, but it's follows the same logic of using lactive feedback loop to generate oscillation. And moreover, many of the proteins in this diagram here, like a PER, like a CRY, CQ1, those are all conserved components in both mouse clock and fly clock. So, and we also found that clock is not only existing in the neurons. Clock exists in almost every organ and every tissue and every cell we examined so far, people examined so far. So this is a recording of PER reporter in the suprachiasmatic nucleus in the brain. You can see here, you can see the oscillation of PER. Now, and here in this video here, these are just fibroblast. And you can similarly, you can see oscillation of PER in the cells. So PER, this circadian clock is in every single cell. Moreover, with the advance of the genome techniques, people have found that thousands of genes in our cells also oscillate with over a 24-hour cycle. So this circadian clock is everywhere in the cell. It affects a lot of genes. And what's the function of the clock? What does the circadian clock do for in terms of human physiology and health? So there are a lot of links between disruption of circadian rhythm with diseases such as increased risk of cancer, mental diseases such as bipolar disease, and a lot and links with metabolic diseases. Here's one example. So this is diabetes and metabolic diseases are more well characterized, very well characterized link. So you can see here the clock mutant mouse here. They have they actually develop diabetes with increased glucose level in their blood and no insulin level in their blood. Now, also, not only and also this mouse tends to develop obesity. And similarly, in humans, studies have shown that if we are working against our own internal clock, it's not the sleep duration per se, but discrepancy between your internal clock and your social clock. That's called social jet lag. That's linked to obesity. So as you can see, as the social jet lag hour increase, the chance of develop obesity increase. So there is definitely a lot of links between circadian rhythm and the disease, but also not only it helps us to understand the mechanism of disease, but it's also quite important for us to understand human behavior. Now, so some of people here are young students, and you may find it's very hard to go to sleep at 11 p.m. as you should be, and it's very hard to wake up at 7 a.m. And some of the parents of teenagers may find it's very hard to wake up your teenagers at 7. So actually, there's a biological basis for that. People have done research using different mammal species. As summarized in this chart here is 6 mammalian species. And during puberty, they actually have a delay of their circadian clock. So there anecdotal evidence only conducted one school, or actually only one school, that if you start the class for teenagers at 10 a.m. instead of 8 a.m., that actually increased their test scores in exam. So actually, I will finish here. And just to, again, as a Drosophila person, Drosophilist, want to emphasize that this is a small research organism that have generated, research on Drosophila have generated 6 Nobel Prize in the past 100 years. And for us, flies and humans are not that different. And I think I will finish here and take questions. Okay.
Thank you for the wonderful talk, and I'm happy to be the first student to ask the question. So my question is, for the aging people, most of them are easily fall asleep at daytime, and then they keep awake at night. So I wonder if this phenotype is a normal phenotype, just like the young children. So just like the young children, they tend to sleep at night. Yeah. Or there are some defect in their here.
So actually, that's a very good question. So there is a link between aging and the circadian rhythm change. So as it's actually established research showing that as people age, they tend to behave more like that per mutant. Instead of sleeping regularly for a long period at night, they tend to wake up several times, and during the day, they tend to take more naps. So it's more similar to per mutant. So in 2015, there is a Cell paper about what's the molecular mechanism, what's the molecular basis for that observation. So people have shown that the SIRTRAN2 level decrease in mouse seems to be the molecular link there. So as we age, SIRTRAN2 level decrease, and that actually is the key link to a degenerating clock.
I wonder, is there any mechanisms about to adjust our circadian rhythm? For example, if I fly to America, and then of course I will suffer from the flight jet for maybe quite a long time, then how can I adapt to the American time after several weeks, 1 week or 2 weeks?
Okay, actually, that's also a very good question. So
I would say that, and I'm very interested in the answer to this question.
So our clock is innate, so it runs even we are placed in constant darkness, but it will be entrained by the night. So every morning, the exposure to the sunlight actually readjusts our clock. So our clock actually is 25 hours. So if we're put in a cave, we will wake up later and later every day. But because we get exposed to sunlight, your clock is reset. So that tells you for a jet lag, the best thing you can do is to get a lot of exposure to sunlight. That will help you adjust to jet lag much quickly. So yeah, there's an entrainment by the light.
Yeah, thank you for the nice talk. I'm not very familiar with fly biology at all, yeah. But I'm curious if you know something about the epigenetics of this process. I mean, you talked a lot about the genetics, and I'm just wondering if you have also something.
So they actually have shown this. So you can see in this, in Takahashi's, I think, 2014 science paper, or 2012, I think actually 2012 or 13 paper, a little bit earlier than 2014. He has shown that those histone markers, the RNA polymerase to posing, those type of epigenetic markers change as during the day. So what they did is they take tissues at different hour and then do the genome-wide profiling. So sounds like they're also at the epigenetic level, they oscillate.
So following on from that then is, if you have a defect in epigenetic regulation, does that then imply that you will also have
potential input from like a yeah, that's I would say yes, and actually don't know the mechanism of drinking coffee or drinking alcohol, how that plays into setting your clock epigenetically. So, but I guess the research is not there yet.
So my question would be, can you walk us through, because when you speak about circadian rhythms in us, virtually every organ system in our body cycles.
Yes.
And yet it all entrains to a similar clock. Our livers don't have eyes. They're not sensitive to light. So can you give us a sense of how this clock per cycle actually then is read? How does my liver tell time?
How should you? Okay. So I will only I don't work on circadian rhythm.
Neither do I. So if you answer wrong, I won't know.
Let me say how what I learned from literature. So the light entrainment comes from the photoreceptors, the neurons that link to the photosensing system. Now that probably will project to neurons that may be secreting hormones, those type of humoral signal that potentially can influence the liver like a clock. So at least I think from what I read, it sounds like this is at least a paradigm in the field.
So Dr. Yen and I will applaud you all for staying awake, but please also join me in thanking Dr. Yen for a great explanation of this year's Nobel Prize in Physiology or Medicine.